At the time the Meselson and Stahl experiment was performed, there were three proposed ways of DNA replication. First, there was conservative replication, in which the DNA molecule stayed intact and the new DNA molecule was a copy of the original, but it did not contain any of the original DNA strand. Second was the semi-conservative method, in which the original DNA molecule separated into two DNA strands. These two strands were used as templates for newly synthesized DNA, giving two DNA molecules which contained both original and new DNA. The third method of DNA replication proposed was dispersive. In this process, the DNA molecule would be cut at various parts and these sections would be copied, then reattached to produce two DNA molecules that randomly contained parts of the original DNA and the new DNA. In 1958, Meselson and Stahl conducted the pulse chase experiment to see which type of DNA replication actually occurs. First, E. coli was grown on a medium containing nitrogen-15. This ensured all bacteria that grew and reproduced contained the nitrogen-15 isotope. When this generation 0 was centrifuged, a single band was formed at the 15 mark. The bacteria was then transferred to a medium containing nitrogen-14. After 20 minutes, a new sample was centrifuged. This first generation produced a single band in between where the 14 and 15 band would occur. After another 20 minutes, a second generation was obtained, again from the nitrogen-14 medium. When centrifuged, it produced a band at the 14 mark, and there was a band again in between 14 and 15. From these results, it was determined that DNA replicated by the semi-conservative method. Generation, the original DNA molecule that contained nitrogen-15 separated. This produced two template strands that the new DNA strand, which contained nitrogen-14, could synthesize from. Overall, the two DNA molecules are produced containing an equal amount of nitrogen-14 and 15. This is why, when centrifuged, a band in between 14 and 15 occurred. During the second generation, a second round of replication occurred in the nitrogen-14 medium. During this replication, the DNA molecules split, producing 4 template strands. And new strands containing nitrogen-14 were produced. Overall, this produced two DNA molecules with both nitrogen-14 and 15, and two DNA molecules containing only nitrogen-14. This is why, when centrifuged, a band at the 14 mark was created, and a band in between 14 and 15 also occurred.